Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below. All right, so this is the last lecture for cytology, and in this lecture we're going to cover the cytoskeleton, the different components and its function. So just an overview, the cytoskeleton, it's a complex network of protein filaments within the cytoplasm of both prokaryotes and eukaryotes. So it's in both types of cells, and you could almost think of it as the scaffolding of the cell. It's the structural support of the cell, just like in a building. But it actually has a lot more functions than just scaffolding or structural support. So sure, it gives the cell its shape. It also helps provide mechanical resistance to outside forces and prevent cell deformation. So if a cell's getting compressed, it can help keep it from getting totally destroyed. It also interacts with extracellular matrix. So outside you have, extracellular filaments and proteins like this, and you can actually have these transmembrane proteins that connect the cytoskeleton to the extracellular matrix. And this actually can serve as kind of the ultrastructure of tissues. It can help bring cells together to form tissues. Because if you remember, first goes cells, then you form tissues, and then you put tissues together and you form organs. The other thing the cytoskeleton can do is it can enable contraction of the cell, which aids in cell division and cell movement. So if you're going to divide the cell, so you can contract, the cytoskeleton helps it contract and break apart into two. It can also help with cell movement. It forms specialized structures such as cilia and flagella, and these can also aid in movement of the cell. The other thing it can do is help facilitate endocytosis, which is essentially when you bring components into the cell. So you can engulf them in a vesicle, and then close that off and bring them into the cell. And so the cytoskeleton can help do that as well. It can also help facilitate intracellular transport of vesicles and organelles. And in fact, some of the cytoskeleton, it can also serve as almost like a subway track or a train track, if you will, in a sense that you're carrying certain cellular components across these tracks, quote unquote tracks, to bring them from one area of the cell to another. And then within the muscle cells, muscle contraction itself is actually performed by the cytoskeletal filaments. So the first component of the cytoskeleton that we'll talk about is microfilaments, which is also known as actin filaments. And these are composed of G-actin monomer proteins that assemble to form polymer. And this is actually a process that involves ATP. So let's say you have the G-actin monomer like this, and then you combine it with ATP like this. So you have ATP then bound to the G-actin monomer, and then what happens is that it joins with an already existing G-actin polymer. So it joins in like that. It would almost be like, so for each of these in the figure here, each of these would be a G-actin polymer. So this is a G-actin polymer. And then this is as well. And so if you have an ATP-bound G-actin monomer, it can then join in with this G-actin polymer. And then what happens is that these G-actin polymers, they intertwine to form F-actin. So these two combine together. You can see these two polymers interacting like this. Together, that's what forms F-actin. And that's the final structure then of microfilaments. Now, these growing G-actin polymers, they enable microfilaments to carry out some of their functions by generating a force when the growing positive end makes contact with an adjacent structure. So let's designate this the positive end here, the negative end here. And so it's going to be growing out from this positive end. You're going to be adding those G-actin monomers bound to ATP to this section here. And so it's going to keep growing out. And it enables then these microfilaments to make contact with adjacent structures, which is really important in processes such as contacting the cell membrane during cell movement, so to help the cell move, or during cytokinesis, when the cell's actually breaking apart and dividing. And so then this ties into kind of the overall functions of microfilaments: cell movement, cytokinesis, intracellular transport, maintaining the cell shape, and then serving as a track for myosin motor proteins as well. So these myosin motor proteins can then move along these and transport other material within the cell. They're commonly found in muscle cells and then in the microvilli of epithelial cells lining luminal surfaces within the intestines and the kidney. And so the way that works is, so let's say we have some epithelial cells like this, right? And they line a lumen like this. If it's just flat like this, that actually limits the amount of surface area available for absorption. So in some of these areas like the intestines or the kidney where absorption is so critical, you actually can increase that surface area by creating these epithelial folds like this within each cell. And so as you can see, that greatly enhances the available surface area available for absorption. Now, within each of these folds like this, you have these microfilaments that help maintain the structure of these epithelial folds and kind of hold them in place so that you can get really solid absorption across. So next we'll talk about intermediate filaments. Structurally, they consist of 8 protofilaments that join end to end with staggered overlap. And that's really, I don't think you need to know much more than that unless your class really goes into detail. But for most exams, you're really just going to have to know that. And then really what's more important is actually the function of intermediate filaments. So they provide structural support for cells, which is just part of being the cytoskeleton. But then they also help with anchoring organelles and then formation of the nuclear lamina and scaffolding. And so as you can see that here, the way they're arranged, they have these long filaments like this, and they're organized in this fashion like this in the figure here. And then specifically, they help strengthen nerve cell axons, which are called neurofilaments. So if we draw that here, if we draw a neuron like this, so a neuron is a specialized type of cell found in the nervous system. And so it has a special shape. So here's the cell body here. And then you have the nucleus here. And then here you have this long extension from the cell body called the axon. And then down here, this part of the neuron is called the axon terminal. And then what happens is that you form a synapse where you have another neuron like this. And this is how messages are conveyed across the nervous system. And then we won't go into any more detail than that. What you really just need to know is that you have intermediate filaments here that kind of help with the structural support of the axon specifically, and these are called neurofilaments. So if you get asked that, what's the special type of intermediate filaments within the axon, the answer is neurofilaments. They also help maintain the structure of muscle fibers, and specifically that type of intermediate filament is called desmin. And then another thing intermediate filaments do is they help contribute to cell and cell-to-matrix junctions. So if you have 2 cells being bound together like this by a junction, and then also, like we were talking about, the extracellular matrix, you can have intermediate filaments in here, and then you have connections out here as well. So the last component of the cytoskeleton we'll talk about are the microtubules. Now the assembly of microtubules is kind of neat, and it's definitely something you could be tested on. And so the way it occurs is you have alpha and beta tubulin proteins that polymerize end to end to form protofilaments that form a hollow cylinder structure. So these are the final protofilament structure here. So this whole thing is a protofilament. And what's important to note is that if you looked at a cross section of this, it's actually a hollow cylinder in here. It's almost like a pipe. Now the way you get to that is you have alpha and beta tubulin monomers like this. So these are each monomers. And then they combine to form an alpha-beta dimer. And then these dimers have to get activated by GTP, so they need an input of energy. So you use up a GTP, and this gives you a GDP. So then you've got this activated alpha-beta dimer. So we'll say activated down here. So then the way you get from here to one of these protofilament structures is then it's got to join end to end with another activated alpha-beta dimer. And so what happens is, let's draw another one over here, is the alpha from one is going to join with the beta from another. And so what ends up happening is It's kind of a repeating sequence. You go alpha, beta, like this, and then so on. And so for each of these, let's say this is an alpha here, this is a beta like this, and then it's joined with another alpha here, and then this guy here is a beta, and it goes on, so on, and so on, until you form this whole cylindrical structure, which is known as a protofilament. Protofilaments are polarized, so they'll have a negative and a positive end. So we'll say this is the positive end here, we'll say this is the negative end here, same thing here, we'll say for this one, this is a separate protofilament, that's a positive end here, negative end here. And elongation can actually occur at either of these ends. So it can occur at the positive or the negative end, but it's considerably quicker at the positive end. The other thing to make note of is that microtubules can disassemble. So you can come from this protofilament and disassemble back into these alpha and beta monomers. This is important particularly for the mitotic spindle, which is important in mitosis for aligning and then also segregating chromosomes in preparation for cell division. We'll talk a little bit more about that later. So some of the functions of microtubules, again, as part of the cytoskeleton, they help support cell shape and polarity. They also help provide tracks for intracellular transport. They help with cell movement. Like I was talking about, they help with cell division. So in mitosis and meiosis. And then they also help with, as part of these processes, chromosome segregation. So something I want to point out real quickly here is that microtubules are typically nucleated by organelles called Microtubule Organizing Centers, or MTOCs, and they actually enable that polymerization in the positive direction that we were talking about. This actually comes into play during mitosis and meiosis. And so again, here's the phases of mitosis shown here in this figure. And these lines right here, these are actually your microtubules. And then here, the centrosome is actually the primary MTOC and is critical in cell division. And so right here, this would be your MTOC, and then same thing here, MTOC. And so these help anchor the microtubules and again enable that polymerization. As you can see here, the microtubules attach to these chromosomes, which then enable them to line up during metaphase, which is critical. And then also, as you can see, the microtubules are retracting or disassembling in each direction here to help pull these separated chromatids to their respective sides of the cell in preparation for cell division. The other thing is that microtubules, they serve as tracks for motor proteins to travel along, such as kinesin and dynein. So kinesin and dynein, they carry out hydrolysis of ATP, so it's an energy requiring process, and it generates energy to transport proteins, vesicles, organelles, and other molecules along the microtubules across the cell. The cover of our biochemistry book is a great illustration of this. So this would be your microtubule here, and then also you can see it back here as well in the background. And then here's your motor protein right here, and it's carrying this large vesicle. And as you can see, it's essentially walking, if you will, in this animation across the microtubule. And it's a pretty cool illustration. You can actually go on the internet and find really cool animations showing this. And so that's why part of the reason we decided to include it for our biochemistry cover is it's just a really cool biochemistry animation. And so it gives you really a sense of that it's literally kind of taking steps across this microtubule track to bring this vesicle from one part of the cell to the other. And again, the two motor proteins you want to be aware of is kinesin and dynein. So let's draw a microtubule like this. So you've got a positive end here, right? And then you got a negative end here. So kinesin, we'll draw one right here. It's carrying a vesicle here. And so this is, we'll do K. It actually moves molecules towards the positive end of the microtubule. One way I kind of remember that is, it's kind of a silly way, is, you have potassium, which is K+. And so that's one way I remember it. Maybe that works for you, maybe not. Just thought I'd pass that on. Then you have dynein. So if we go down here, it's carrying a vessel. This is dynein, D for dynein. This actually moves towards the negative end of the microtubule. So kinesin will carry material towards the positive end. Dynein carries material towards the minus end. All right, so we'll close this out with a little clinical pearl, or actually pharmacology tie-in here. So there's a class of drugs called microtubule inhibitor chemotherapy drugs. And some members of that are paclitaxel or Taxol, vinblastine, and vincristine. And these affect microtubules during the M phase of the cell cycle. So remember the cell cycle, we'll draw it up here. You've got G1, You've got the S phase, then you got G2, and then you got the M phase here, where essentially you're going through mitosis. And so these drugs specifically, this would often a test question, especially on a board exam, is which phase of the cell cycle do they affect, and it's going to be the M phase. So there's two mechanisms here that these drugs carry out to affect the structure of microtubules and their function within the cell, and specifically during cell division or mitosis. So the first type is hyperstabilization, and this is kind of an indirect way of actually affecting the cell function and actually causing apoptosis. So what happens is that you cause hyperstabilization of polymerized microtubules. So let's take this right here. You'll have the drug bind at a polymerized microtubule like this. Now, like I said, during anaphase, you're trying to pull these chromatids apart and try to carry them to either side of the cell in preparation for cell division. To do that and to carry out cell division, you need the microtubules to actually disassemble and so to come apart. However, by this drug binding to the microtubule spindles, it prevents this. It prevents them from breaking down and actually keeps them stabilized. And so it interferes with the cell division process. And so by doing that, by essentially stabilizing these microtubule structures and preventing their needed breakdown, it actually triggers apoptosis. And so this is actually the mechanism of action of paclitaxel. The other mechanism is that for vinblastine and vincristine is they actually bind to beta tubulin. So remember, you have your alpha and your beta, and they come together and they form your alpha beta dimer like this. So what happens is that the drug will bind to, just draw this red dot here to symbolize the drug. It binds to the beta tubulin and it inhibits this process. So it inhibits polymerization of microtubules. So it prevents this mitotic spindle from even forming in the first place. And then that's another way you can prevent cell division. And so this is really important because you can get kind of tripped up on tests. So you got to remember that they don't just affect microtubules, you got to remember how they affect. Is it hyperstabilization or is it inhibiting polymerization?
Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.